Welcome to Let's Learn Plants. I'm Riddy, and I'm so excited you're joining me today. We are going to embark on an absolutely incredible microscopic rescue mission right inside the lab, extracting the hidden molecular machinery that literally makes plant life possible.
So today's explainer is all about the journey of a protein. Think of this whole process as a highly technical obstacle course where we need to rescue a single, specific target protein that's trapped inside a remarkably tough plant cell. It's a multi-step process, and each technique we're exploring today acts kind of like a specialized tool designed to clear a very specific biological hurdle.
Okay, let's dive into this. Here is our macro to micro lesson plan for isolating specific plant proteins. We'll start with the dynamic proteome, then protein extraction, salting in and out, cleaning up with dialysis, protein chromatography techniques, and finally, high-performance liquid chromatography. Section 1. The dynamic proteome.
All right, before we can even begin our extraction mission, we have to zoom out and look at the entire cell's protein landscape. Because our target protein, well, it's just one tiny piece of the proteome. By definition, the proteome is the entire complement of proteins expressed by an organism, cell, or tissue at a particular time. Notice that crucial phrase, at a particular time, right? Unlike a plant's genome, which is basically a static, unchanging instruction manual, the proteome is the actual functional expression of those genes. It is immensely complex, and it's literally always changing.
In fact, you can think of the proteome as a moving target. It fluctuates constantly based on three specific factors. First, the cell type. A leaf cell's proteome is going to look vastly different from a root cell's, right? Second, the developmental stage. Are we looking at a tiny embryo or a mature adult plant? And third, environmental conditions. Things like shifts in pH, temperature, or the sudden presence of hormones and nutrients. Because of all this, when we go hunting for our target protein in the lab, getting our timing and location exactly right is absolutely crucial.
Section 2. Protein extraction from cells.
Okay, so this is where we actually step up to the bench. This is the messy, physical process of breaking our delicate protein out of its incredibly tough plant cell wall. We follow a very standard lab protocol here. Step 1 is lysis. We literally bust open the cell membrane using physical force, like sonication or chemical detergents like Triton X100. And this creates a sort of messy cellular soup containing basically everything that was inside the cell. To clean it up, we move to step 2, which is centrifugation. We take our soup, put it in a machine, and spin it incredibly fast. The heavy, insoluble debris gets slammed to the bottom of the tube to form a solid pellet, while our precious target protein stays floating safely in the clear liquid above, which we call the soluble supernatant.
But remember, we are working with plants here, and plant cells are notoriously tough, requiring some vital extra steps. We frequently have to freeze the tissue solid and grind it up in liquid nitrogen just to turn it into a powder and instantly deactivate these dangerous enzymes called proteases that would otherwise completely chew up our target protein. We also deploy high salt buffers to smash through those rigid cell walls, and we have to add special inhibitors like PVVP to stop harmful plant phenols from damaging our catch. So yeah, it requires extreme measures, but honestly, that's what makes plant molecular biology so fascinating.
Section 3. Salting in and out.
Let's move to our next step and see how this builds. Once we've successfully extracted our soluble cellular soup, we introduce our first chemical technique for separating our specific protein from the rest of that mixture. This is a brilliant balancing act called salting in and salting out. When we add a low concentration of salt, that's salting in, the salt ions actually shield the protein's natural surface charges. This stops the proteins from clumping together and significantly increases their solubility in the solvent. But if we blast the solution with high ionic strength, typically using ammonium sulfate, we get salting out. Paint a mental picture of this with me. The massive amount of salt intensely competes with the proteins for water molecules. The proteins actually get stripped of their hydrating water shell, which forces their hydrophobic cores to interact. They clump together and physically precipitate right out of the liquid. Since different proteins precipitate at different salt levels, we can strategically use this to just drop our target right out of the solution.
Section 4, cleaning up with dialysis.
So we've successfully precipitated our protein, but now we have this heavily salted solution that needs to be washed before we can proceed. To fix this, we use a remarkably gentle technique called dialysis. Dialysis removes small molecules, like that ammonium sulfate we just piled in, using a semi-permeable membrane. Think of it as a highly specialized microscopic net. We place our salty protein solution inside a dialysis bag with a specific molecular weight cutoff. Then we just immerse that bag in a large bath of dialysis buffer. Simply through the power of diffusion, the tiny salt molecules escape through the membrane's pores out into the bath, while our large, precious target proteins are safely retained inside the bag, perfectly desalted.
Section 5, Protein Chromatography Techniques.
And this brilliantly illustrates the most precise stage of our obstacle course to completely isolate our target. Our first precision tool is gel filtration, also known as size exclusion chromatography. Imagine a column as an obstacle course filled with inert, porous beads. We pour our desalted protein mix into the top. Now, counterintuitively, the largest proteins actually win the race here. They're far too big to fit into the microscopic pores of the beads, so they completely bypass them, traveling straight through the void volume, and they elute first. Meanwhile, the small proteins kind of get trapped wandering through the inner volume of all those tiny pores. They take the longest path possible, and therefore they elute last.
Next up is ion exchange chromatography, or IEX. Now, this technique doesn't care at all about size. It separates based purely on the net electrical charge of the protein relative to the buffer's pH and the protein's isoelectric point. We pass our sample through a column packed with either positively or negatively charged functional groups. It's essentially a game of electrostatic interaction. For example, negatively charged proteins will stick tightly to a positive anion exchanger, while neutrally or positively charged proteins wash right on through. Once our target is stuck to the column, we force it to release by flushing the system with a heavy salt solution that competes for the bond, or by tweaking the pH. Then we have the most precise technique of the bunch, affinity chromatography. This relies on intense biological specificity. It's exactly like a lock and key. The stationary phase is loaded with a specific ligand that only our target protein recognizes and binds to. A really great example is IMAC, where we use immobilized metal ions to grab proteins that have been engineered with a specific His tag. While the target protein binds tightly to the metal, literally everything else washes away. To collect our pure sample, we perform a competitive elution, flooding the column with something like imidazole, which kicks our protein off the binding site and right into our collection tube.
So the crucial point is really how these distinct principles work together. To summarize, gel filtration separates by size, using those porous beads. Ion exchange separates by charge, by manipulating the buffer's pH against the protein's isoelectric point. And affinity separates by specificity, using that lock and key ligand binding. Honestly, mastering how these three distinct principles work together gives you the power to isolate almost any protein imaginable.
Section 6, High-Performance Liquid Chromatography. This brings us to the ultimate, most advanced analytical technique in our journey. HPLC takes the chromatography principles we just learned and massively upgrades the hardware. In HPLC, we use a remarkably finer stationary phase, with uniform particles as tiny as 3 to 5 micrometers. Because the column is packed so incredibly densely, gravity simply isn't strong enough to pull the liquid through. So we have to utilize extreme high-pressure pumps. We're talking pushing at 5,000 psi or more to forcefully drive the mobile phase through the system. This extreme pressure yields amazingly fast analysis speeds and superior high-resolution separations. It's especially useful in reverse phase HPLC, where we separate tiny peptides based strictly on their hydrophobicity.
Well, we've made it through the obstacle course. But before we wrap up, I want to leave you with a real-world challenge to think about. How could you apply these precise purification techniques? You know, the freezing liquid nitrogen, the careful dialysis, the highly targeted chromatography, to discover a brand new, highly active enzyme hidden inside a tough, resilient desert plant? The exact step-by-step tools you need are right here, ready for you to use in the lab to make your own molecular biology discoveries. Keep questioning, keep exploring the microscopic world, and I will see you in the next explainer.